Hello everyone, my name is Daniel Osorio. I'm a postdoctoral fellow at the Livestrong Cancer Institute at the University of Texas at Austin. And today I'm going to present you a project I have been working on the systematic characterization of the gene regulatory network's rewiring patterns mediated by non-coding regions perturbations. The idea is that we are able to construct an individual cell gene regulatory network to study how the regulatory mechanisms change when there is a non-coding region perturbation. So to construct an individual cell gene regulatory network reconstruction, currently the most used tool is Scenic. Scenic has been cited more than 1,600 times, and it takes into account certain assumptions that are not totally biologically accurate. For example, it only works with transcription factors that are seen in the gene expression matrix, and it is the correlation metric that they use is positively defined since they work with random forests and agglomeration of trees, and the transcription factors of the activity of the transcription factors is returned as a binary matrix where the transcription factors is active or not. So this is in disagreement with the biological reality because the transcription factors are typically lowly expressed. They are hardly seen in the gene expression matrix of single-cell RNA-seq data because the large amount of dropouts that we see in that data type. The transcription factors also can be activators or repressors of the gene expression of the genes, and then this requires a metric that allows for positive and negative values. And also the activity of the transcription factors is dependent on the concentration that we observe in the cells. So the most of the transcription factor, the most of the activity in there. And this is something that is missed in the output of Scenic. So in order to account for those changes, we recently developed a tool called Scorpion. Scorpion stands for Single Cell Oriented Reconstruction of Panda-based Individually Optimized Networks, and it's an implementation of the Panda algorithm from single cell RNA-seq data. So the Panda algorithm is an optimization algorithm that uses message passings to integrate different data types. In this case, protein-protein interaction, transcription factor motif in the promoter region of the genes, and the single cell gene expression data. So the correlation structure of this gene expression is taken into account to update or integrate all the information together to generate a refined gene regulatory network that ensures the agreement of all the biological data that we have access to. So in Scorpion, we are able to assess the activity of all the transcription factors, even if it is absent. And we ensure that the metric that we use accounts for the directionality of the regulation. So transcription factors can have an activator activity or a repressor one. Also, transcription factors are accounted to be concentration dependent, and we use a continuous metric for that. And we also account for the cooperativity of the transcription factors by the information that is provided by the protein-protein interaction. So the first thing we did with the Scorpion was to evaluate its performance. And for this purpose, we use a tool called BLine. BLine is a tool designed specifically to test the performance of gene regulatory network construction algorithms. And by using BLine, we were able to compare the performance of Scorpion against 12 other algorithms. So in the plot on the right side of the slide, you guys can see that Scorpion outperforms all the other 12 algorithms, and it provides the higher precision recall curve metric with an increase of at least 20% of the performance. And compared with others, Scorpion is also faster, and it also provides more information about the motifs that are present in the networks. So because the information provided by BLine is too small and don't recapitulate a real biological experiment, we decided to apply Scorpion on two different experiments that contain transcription factor perturbations. The first one is a double knockout of two transcription factors that define the identity of the enterocytes, the HNF4 alpha and gamma transcription factors. When we construct two networks for each one of the cases, one for the wild type and the other for the knockout, we found that there is a shift on the weights associated with these transcription factors, and those shifts are consistent with the directionality that was targeted in the wet lab. So there is a downregulation or a decrease in the weights that we see in those weights of the transcription factor and target gene interactions. And when we compare the differences in the specific target genes, we found that the top ones at the bottom of the list, the most downregulated ones, are in fact associated with the markers that define the identity of the enterocytes. This confirms the authors of the dataset claims and also confirms the ability of Scorpion to identify changes in the transcription factors and the specific target genes. To account that Scorpion is also able to identify upregulations of the transcription factor activity, we use a recently published dataset that includes stem cells treated with the overexpression of the DUTS4 transcription factor. And by doing this, we construct also two gene regulatory networks, one for the wild type and another for the overexpression of this transcription factor. And when we compare the weights, we found a shift that is also consistent with the experimental design. The shift is in the positive side, so all the weights are larger than they were before of the overexpression of the transcription factors. And when we compare the differences in the effect over the target genes, we found that the ones associated with the identity of the mark of the AC cell-like are in the top of the list. So they are in fact upregulated. So by using this information, then we decide to use another tool that was developed by my QJ called lioness. And when we combine Scorpion with lioness, we are able to construct an individual cell gene regulatory network. So these individual cell gene regulatory networks are important because they allow us to associate different covariants of the information. So in here in this slide, we see the network zoo, that is a family of tools developed in the Broad Institute that accounts for different techniques and different experiments that can be done over the networks that are constructed using the Panda algorithm. And it also displays the formula that is used to construct an individual cell gene regulatory network. So to test the performance of this combination of tools, we decide to apply it over this dataset, the GSE 185995. And this dataset was designed to account for the non-coding regions perturbations of the human genome in breast cancer cell line. So the experimental design is that the authors of the dataset generated a set of single guide RNAs, and then they infected the cells, and the guide RNA will cause a perturbation in the non-coding region of each one of them. And sadly, there is no individual association, so we individual perturbations of one guide RNA by one cell. There is a combination that approximately 30 guide RNAs per cell in at least 50,000 cells. So we use this information and generated individual cell gene regulatory network for each one of them. Then we moved to that analysis, and we found that in the dataset after quality control, there are 54,000 cells, and the distribution of the number of guide RNAs per cell are displayed in the panel A. In the panel B, we have the distribution based on the UCIS feature types, and we see that most of the perturbations are located in known regions or not very well characterized. The others are in the promoters and in the enhancer regions. And we also evaluated the differences of the transcription factors motifs in each one of the promoter regions, and we found that the top 5 more affected will be SP1 and all the others displayed in the panel C. When we take into account the in-degrees of the network, we use this information to construct a low-dimensional representation of the data. And this is displayed on the panel D. As you can see, the phenotypes that we observe after the perturbation converge to a limited number of phenotypes that are defined for each one of the clusters. And as you can see at the top of the panel D, there is a cluster in dark blue. It means that those cells, we hypothesize that those are the wild type group because they are close to not having any guide RNA on it, and the effect of the phenotype is defined in them. So the second step that we did was to identify the associations of each position of the perturbation in the x-axis in this plot, and the association with the changes in the transcription factor activity on the y-axis. So as you can see here, the majority of the associations that we see are in blue. That means that when the when the non-coding regions are perturbated, the effect of the transcription factors is downregulated. And this is consistent with the idea that the authors of the dataset are targeting by targeting the flanking regions of the well-defined enhancer in cancer, and others that are labeled in red, that are the minority, are known as insulators, and those are associated with the downregulation of the activity of the transcription factors. But when you perturb them, of course, they will allow the cells to overexpress them. So we tested each perturbation and identify what transcription factor will be the most affected one. Then we move to identify the effect of the perturbations over each one of the target genes, and we identify a set of genes that are often more frequently affected by these perturbations. We focus on identifying and characterizing each one of the perturbations positions based on different epigenetic markers that are shown in the range of this plot. And also we define each one of the links weight and the different types. So in this way, we are able to know which perturbation in which regions of the genome are associated with differences in the in-degree of the network when it happens. So we then were interested in to know if there was an association between the position of the perturbation in the x-axis and the position of the transcription start site for each one of the genes. We found that there is not a direct association of a guide RNA target of guide RNA perturbation and the more affected gene. So not because we perturb the closest non-coding region of a gene, we will have that gene specifically as a significantly perturbed. And we found that still there is an enrichment of the cis regulatory mechanisms, but this one is not that highly different to other trans regulatory elements. So this is important because we found that even though the promoter region is the most important or the most important section of the genome to modify the expression of the genes, the unknown regions that are still not very well characterized are ranked as the second one, and there is no statistical difference in their differences of activity. So we see that the enhancers are not that effective on regulating them, but they are not very well defined for this kind of cell line. So for that reason, we wanted to investigate further, and I develop a new data set where we can actually identify causal associations of the network rewiring patterns. So because this data set that we used before does not have enough cells with a single guide RNA on it, it makes it difficult to model the associations between each one of the perturbations and each one of the rewiring patterns that we see in the networks. So we decided to generate a new CRISPR screen on single cell transcriptomes using crop-seq and use the same cell line but to target individual perturbations in each one of them. So we sequenced 18,000 cells that some of them were removed after quality control. Remove the cells with mitochondrial content above the 10%, and those that fail out of the 95% confidence interval for the regression against the number of genes based on the level of expression. And we identified 1,200 cells carrying a single guide RNA that allow us to test the perturbation effect of 48 defined enhancers. So we generated the figures that are displayed in the panel A to display the mitochondrial content, in the panel B to identify the number of genes per cells, the number of guide RNAs per cell in the panel C, and the similarity between the genes, between the guide RNAs that are observed in each one of the cells. As you can see, there are two main clusters that contain cells carrying a single guide RNA. So these two are suitable targets that induce the proliferation of the cells, and they are interesting to understand how these perturbations regulate this proliferation. So as a conclusion, we introduced the Scorpion algorithm, that is an implementation of the Pan-gene regulatory network reconstruction algorithm for single-cell RNA-seq data. We showed that we constructed individual cell gene regulatory networks under different CRISPR perturbations. We found several non-coding regions in the genome that disrupt the transcription factor activity and the expression of the target genes. We found an enrichment of 6 significant associations between perturbations close to the gene's promoter regions, and we found differences in the rewiring patterns between different feature types, emphasizing on the importance of characterizing the genome non-coding regions. And that is all for today. Thank you for having me.